Right, so today I just wanted to talk to you about some of my recent work looking at the experimental decay of the penis worm Priapulus and its implications for understanding of Burgess shale type preservation and some of the evolutionary implications that it has for interpretations of Cambrian fauna. So in this session, the Cambrian explosion doesn't need much of an introduction, but needless to say, it is a unique event in the history of life on Earth when a large number of phyla simultaneously appeared, animal phyla, at the middle and early Cambrian. One of the reasons we have such a good insight into this evolutionary event is the soft tissue preservation that is characteristic of many fauna from that period of time, including, for example, we've just seen in the previous talk, but more typically as well from the Burgess shale itself and from Chengjiang in China. So given this circumstance of soft tissue preservation, we need to ask ourselves about the mechanisms by which those soft tissues are preserved. And this is quite a mature field, which a number of people have worked on for some time, with these examples here from Butterfield and Gaines et al., whereby we are developing a good idea that we have this recalcitrant carbonaceous cuticle or other macromolecules that are generally resistant to decay and are preserved via an organic pathway by just virtue of their ability to survive decay processes. We also, that doesn't rule out the possibility that more tissues like guts and muscles could be preserved through other fast pathways as rapidly decaying, orthogenically mineralized through bacteria action. And that's something we saw again with the previous talk. We talked about phosphatization. So we have this good understanding of the model, but one of the things we might want to ask is if there's empirical support for this. This is especially relevant when we start thinking about some of the recent fascinating fossils that have been described of arthropods and other ecdysozoans of their internal preservation of neural tissues, which have been used to develop interesting theories relating to the evolution of this important character and also the relationships of arthropods themselves within them. But this raises an interesting question because these are internal tissues that aren't part of that classic cuticular or style. So for this, we might want to investigate the time scale is important here. What tissues are decaying fast, what tissues are decaying slow, and are there likely scenarios which are compatible with Burgess shale type preservation for these brains and vascular tissues? So Ma et al. Demonstrated quite convincingly that the internal tissues are typical of organic style preservation. These are carbonaceous films that have various different levels of diagenesis, which again is typical of Burgess shale type organic preservation. So what about the time scale of these things? Is it likely, given our understanding of Burgess shale type preservation, that we can have preservation of internal tissues that are not of that typical macromolecule or cuticle style? So to test that, I investigated the decay of Priapulus. Priapulus, or Priapulida, is representative of Priapulida over here, which is part of the Scelidophora, which are sister taxon to all other ecdysozoans. They're pretty much largely agreed to be a very good representative for the early condition of the group. So in the first hypothesis that we want to test here is, are patterns of decay consistent with Burgess shale type preservation? And what implications do patterns of decay have for our understanding of the preservation or possible preservation of neural tissues, or well, non-cuticular tissues? And the second is to more classically align with some of my previous research with team in Leicester, is to think about whether or not the patterns of decay undermine phylogeny. What does that systematic taphonomic biases have on our ability to draw evolutionary conclusions from those fossils, which is the game we're in, really. So to do this, I went to the west coast of Sweden, to the Sven Löfven Center for Marine Infrastructure. It seems to change its name every week or so, marine biology, marine infrastructure. And here we used a ship to trawl the benthos of the Gulmar Ford for 5 days, and were able to recover about 20-25 specimens of Priapulus. And here we took I took a character-based approach following previous work on vertebrates and other fauna, looking at trying to focus upon how individual anatomical characteristics decay during that profile. So this includes specimens in seawater, but also specimens buried in sediment. That is just the natural sediment of that they're found in the fjord around them. So this is a simple model, but as I think Duncan Murdoch has explained in the past, we can build in complexity of these models by trying out different experimental variables through increasing complexity of those experiments. So here is a time-lapse photography. This is just over 6 days of a Priapulus decaying. So as you can see, changes are extremely rapid, especially with respect to body dimensions. This is largely related to contraction and decay of the muscles, but the changing of the body shape. But one of the interesting patterns I wanted you to see here was how the cuticle, the inner cuticle, strips away from the outer cuticle, and the inner body shrinks, and that can create, so there's this rapid decay of the dimensions of the body, and that can create this sort of shell or sort of ghost inside the body of where the outer body used to be. I did have some other videos here, but I'm not sure if they're going to work. Let's have a look. So there's another one typically showing that with the destruction of the wastage of the internal anatomy. And finally, this one here, again, showing a similar sort of pattern. Nope, doesn't like that one. Fair enough. So this is that data summed up in a graphical format, whereby we can look at a number of different specimens if they've sort of undergone contraction and peeling away of the inner layer and the outer layer, swelling and contraction and changing of body dimensions in a very rapid sense. This is over a period of 65 hours after death. When we look at the internal tissues, we again see quite striking results. The internal tissues, such as the muscles and the gut, decay very, very quickly and are absent almost completely within a matter of days. And that's true of both the specimens decayed in sediment and the specimens decayed just in seawater. So what does this mean when we try and think about individual characters? So here are the individual characters of Priapulus laid out and ranked according to their decay profile. In this chart, we can see that the vast majority of specimens are completely pristine throughout the entire course of this experiment. So this is intensive sampling for 6 days and then a return visit 6 months later to see what's happened. And because of the timescale, that's actually quite appropriate because almost nothing happens for that 6 months with respect to the cuticular characters of the scalids. The internal anatomy, on the other hand, the non-cuticular characters are extremely decay prone and are lost within a matter of days, almost completely, with the exception of the gut in some cases. So there's a striking pattern here with respect to cuticular versus non-cuticular characters, which is, I guess, is not that surprising. But when we apply the concept of stemward slippage, this idea that there might be decay biases, there's no correlation at all between the phylogenetic informativeness or the diagnostic rank of a character and its position in the decay profile. But that's, again, intuitively makes sense because a lot of the characters here, the scalid characters, are informative down to the genus level. And the scalids are extremely decay resistant. So we don't see anything like that we do for chordates. So to sum up those results, the decay patterns include body dimension distortions, which are extremely pronounced. We see rapid internal non-cuticular decay of anything on the inside of the body, and we see an absence of stemward slippage. So let's put this in context and compare that with Murdoch et al., who did a series of decay experiments on onychophorans. And they find those exact same 3 patterns. There is body dimension distortions, there's rapid internal non-cuticular decay, and there's an absence of stemward slippage. So when we put that in a phylogenetic context and apply the idea of comparative taphonomy in the sense that we might do comparative zoology and try and reconstruct the sort of ancestral condition for this taphonic patterns, we can see that this is, because we don't have any data for tardigrades and nematodes and chinorhynchs, we can make, as we currently understand it, we can generalize this pattern for all early ecdysozoans, including the arthropod stem. So with that in mind, what implications does this have? Well, let's turn firstly to preservation. The cuticle was extremely robust to decay after 6 months with or without a sediment. And this is very consistent with Butterfield and Gaines et al., where the organic pathway of BURGESS shale type preservation is related to decay resistance. On the other hand, internal anatomy non-cuticular characters decay extremely rapidly, very, very, very fast. So what does that have an implication for other kinds of preservation we see in the BURGESS shale? So returning back to the situation of neural tissues and brains, given our understanding of decay on this timescale being extremely fast, organic preservation of unpigmented non-cuticular anatomy is unlikely given current BURGESS shale type models, at least with any kind of fidelity. Now that doesn't mean that any of those tissues aren't neural tissues, of those patterns that we see in those arthropods. It just means that there's an inconsistency here between our understanding of decay, BURGESS shale type models, and the anatomy we see in them. So a suitable null hypothesis that we'll need eliminating in many of these instances is that any internal structures don't actually represent with fidelity the internal anatomy of what you're looking at, but instead a shrunken internal mass given the separation of the outer cuticle and inner cuticle that occurs in both Priapulids and onychophorans. So in many of those cases I showed you in that slide with all the different fossil brains and vascular tissues, that's very easy to do. Some of the anatomy is quite complex and it will likely stand scrutiny. Others is not so sure. Either way is a null hypothesis that will need eliminating in order to be able to account for patterns of preservation and decay. So moving on from that, what are the implications for phylogeny? Well, using this phylogeny from Wills et al. Of Scelidophora and taxa, they construct a preapulid crown group, including the living forms, and a preapulid stem group. So I went through their data set and identified the characters that might be taphonomically unlikely to be preserved with fidelity, those that the decay experiment have revealed to be extremely changeable. That's the body proportions and the presence or absence of non-cuticular anatomy. When those characters are stripped away from organically preserved fossils, not all fossils, just those that we have a consistent understanding of organic preservation, this completely transforms our shape of the phylogeny. So it initially looks subtle, but the implications for it are massive. Instead of these fauna, this large Cambrian fauna containing all our favorite classic ones like Otoya, are all on the preapulid stem, which is what we would understand. If we take out those potentially taphonomically ambiguous characters, they all sit on the stem to this group of loriciferans and chinorhynchs instead. So these characters, although small, are having a massive impact upon the phylogenetic implications and our evolutionary inferences that we draw from those fossils. When we do the same thing with arthropods, using the data set, or panarthropods I should say, because there's quite a lot of different phylogenies out there, we find no impact at all. The neural tissues are not changing the shape of the tree at all. So, and just as a little Christmas present, an unexpected story for this was its implications for chordates, which I didn't expect to find at all. So, for those of you not aware, preapulids have that typical vermiform circular musculature, or we're all the same. But through the decay process, they transformed into sigmoidal segments. And this is a typical characteristic that's been used to infer chordate characteristics for Tully monster or for Palaeontological. But actually, this would suggest here that because this chordate or supposed chordate characteristic can be replicated from a non-chordate pattern, that caution should be applied when invoking segmental segmentation as a chordate affinity. Instead, we should stick to the classic chordate characteristic, which is sharp V shapes, W shapes, or Y shapes. So, in conclusion, patterns of ecdysozoan decay are consistent with models of Burgess shell type preservation. The non-cuticular characters tissues are currently incompatible with models of Burgess shell type preservation, and at least in the simple way that we understand them, and that the phylogeny has been demonstrated to be sensitive for that for preapulids, but not for arthropods. And then finally, sigmoidal segmentation is not a reliable indicator of chordate affinity. So thanks to Zhao Yamar and Sarah Gabbard and other people that I've spoken to about this, and funding from NERC and the BBSRC. And if you want to find out more about this, this paper came out earlier this year in Scientific Reports. Thank you for your attention.